Good morning. I want to talk you through today meiosis, which is the second kind of cell division. But before we go to that, I want to show you this slide just to remind you where we were, right? We just finished up mitosis, and in mitosis, we take one diploid cell and we create two identical daughter cells from that. You can see over here I show a kidney cell and it's just completed division, so you can see two identical copies here. Today, I want to walk you through meiosis, which is the second kind of cell division. And in this particular kind of cell division, we are going to start with one diploid cell, but this time we're going to go through two rounds of division so that we end up with 4 haploid daughter cells. These cells will be unique. And over here, I picture a sperm and egg. This is the gateway to sexual reproduction. So we use meiosis to create these sex cells. All right, before we begin, there are a few vocabulary terms that I think you need to know in order to understand this. So over here in this picture, I have a karyotype, and a karyotype is where we take all of the human chromosomes and we actually link them up by their length. We then group them into sets. And so you can see chromosome number 1 is very, very long, all the way down to chromosome 22, which is very, very short. And you can see that we can arrange these particular chromosomes inside the human cell in matching pairs. Now, it's interesting. Each person contains 2 chromosomes that hold the same sets of genes. These are known as homologous chromosomes. I'm going to ignore this 23rd set just for a second and I'll come back to it. It is a mismatch set, but all the rest of them are matching. And you can see these different regions show different genes that match between these chromosomes. Now, where did these matching sets of chromosomes with the same sequence, where do they come from? Well, one chromosome from each set comes from an egg, the female, right, will donate it, and one chromosome from each set will come from the sperm or the male. So essentially, one of your homologous chromosomes comes from your mother and one comes from your father for each of these sets. Now in humans, this is interesting because our diploid number is 46. And so that is where you have 2 of each of your homologous chromosomes, one from your mom and one from your dad. But haploid number is 23. So if we half that diploid number, we get 23. And so in order to conserve or keep this number 46 from one generation to the next, we have to have a haploid number for our sperm. Our sperm have to bring half of the chromosomes to the table. The egg has to bring the other half of the chromosomes to the table so that at fertilization, we get that 46 chromosomes back, right? And we don't change that number. So in humans, this is the way it works. And meiosis is the process that's going to allow us to create these sperm and eggs with half this original number. So that's why it's an important process. Last, kind of bit of vocabulary or just background, these chromosomes out here, 1 through 22, that come in these matching sets, homologous chromosomes, where they are the same length, are known as autosomes. This 23rd chromosome, which is often shown as a mismatch set, like in this picture, is known as the sex chromosomes. And sex chromosomes help us determine our genetic sex. I want to pause and say that gender is a constraint of many different things, including genetics and environment, society, how you're raised, religion, etc., right? But in genetics class, when we talk about our sex chromosomes, we are talking about this 23rd set, and we are talking about the length of it. So you can see down here, X is really quite long. It carries some genes that have to do with primary and secondary sexual characteristics, but also some other stuff like color vision, right? That has nothing to do with your primary or secondary sexual characteristics. Y, on the other hand, is this short piece over here that only what we would consider genetic males to have. And so up here, we will often write males as XY. They contain one long and one short 23rd chromosome. And females will often be written as XX, one long, a second long. So they have 2 long chromosomes right there. All right, so keep this in mind. Autosomes and sex chromosomes, you have 23 total sets of these over here in our karyotype. All right, so in meiosis, we have 2 rounds. So we're going to say that meiosis 1 is round 1, meiosis 2 is round 2. So up here on my screen, I have meiosis 1. Before meiosis 1, we have 2 phases that work identically to mitosis. S phase DNA replication occurs, we copy our chromosomes. And in G2, all of the machinery is prepped for division. Now, we can start meiosis 1. Meiosis 1 is going to have the same order of steps. They're just going to have a 1 after them. So prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase and cytokinesis 1. And then we'll do the same thing for round 2. Right away in prophase 1, in meiosis 1, you have something that happens that is very different. It's called crossing over. This is where the homologous chromosomes line up in tetrads. You can see over here we have two blue chromosomes and 2 red, and they have lined up next to one another. And they go through a process called crossing over. And in crossing over, enzymes run along these homologous chromosome sets, and they cross equal lengths of DNA from what would be your paternal chromosomes and your maternal chromosomes. This particular process allows us to mix up our DNA and create something that we haven't seen before. Now, once you are done with crossing over, enzymes will check this particular process and you will move on to prometaphase 1. And prometaphase 1, the nuclear membrane disappears, and this time your tetrad sets, those sets of 4 homologous chromosomes, link up to your spindle. Once they have linked up, they will be pulled around back and forth until they line up in the center of the cell. This is where metaphase 1 happens. You can see my tetrads over here, these sets of 4 homologous chromosomes in a line. These chromosomes are randomly sorted into the middle of the cell so that you can have some that are blue and some that are red on either side of that metaphase plate. Anaphase 1, your homologous chromosomes are going to split, and so those tetrads split so that one of the homologous sets gets pulled to one end of the cell and the other goes to the opposite end of the cell. In telophase 1, your nuclear membrane is going to return, and cytokinesis, the rest of the cell contents will pinch off. In meiosis 1, we have several things that happen that is pretty different than what we saw in mitosis and pretty different from the second round of meiosis as well. In prophase 1, we have crossing over where we're kind of recombining the chromosomes in new ways. In metaphase 1, we have our chromosomes lining up as tetrads, not as sister chromatids like what we saw in mitosis. In anaphase 1, we have those tetrads separating. And finally, in telophase 1 and cytokinesis, we have our cells splitting, the nuclear membrane reforming, but this time we don't do DNA replication. So we're gonna have a short pause as your cells kind of check themselves, but then we will go forward. So meiosis 2 happens a lot like mitosis. In fact, really what you're starting with and ending with is different, but the phases are much the same. So in prophase, the sister chromatids, they're now not called chromosome sets because they're not identical, but these chromosome sets will find each other and link at the centromere. In prometaphase, the nuclear membrane will sort of unfold and be removed, and then the spindle will grow out and attach to these chromosome sets. In metaphase 2, the chromosome sets line up in the middle. You can see they're sets, they're not tetrads now. And anaphase 2, those chromosome sets separate. And in telophase 2, the nuclear membrane returns cytokinesis. Each of these sort of original single cells will divide their cell contents to create 2 cells. So you can see by the end, we're actually creating 4 haploid cells. Remember, this particular cell started with 4 chromosomes, or a diploid number of 4, and now by the end of this process, we have 2 chromosomes in each cell. Now, that's all I want to say about mitosis and meiosis. Next time we will compare each of them.